So my talk is going to be about my research field, which is translational oncology, which is basically just translational cancer research, and I'll explain what the translational part of that means in a minute. So I've really rather facetiously titled it "Bridging the Gap Between Research and the Real World," because, as I will explain, for me, and the reason I'm so passionate about this field is because it's all about having an impact and changing something in the real world for science, because science can feel very, very abstract at times when you're doing your undergraduate degree, when you're doing your A levels and your GCSEs. It can feel very, very abstract. And when you get to a doctoral level or higher, you can then begin to notice that you have a chance of making an impact with your work, and that makes it really fulfilling. And that's why I'm really interested. So I'll explain what I mean by all that in a minute. So to start with, to give you a bit of background of who I am and what my career trajectory is and where I'm at the moment, we'll go through this slide. And I can't see the chat, by the way, while I'm doing this. So if you've got any questions, we'll wait till the end. So if we start this, we'll do it bit by bit. So I started off at a comprehensive school. I did my GCSEs there. I come from Essex originally, which is east of London. And then I moved to a grammar school, did my A levels there, so biology, chemistry, and maths at A level, and economics at AS level. Then I did my undergraduate degree, which was biochemistry. I did that at King's College in London. Whilst I was there, I was lucky to complete, I worked hard to complete 3 different research placements. So usually as part of an undergraduate science degree, you'll have to do 1 research placement for your dissertation project, in which you'll have to write up a dissertation, which is a long form research summary essentially. So I did that, but I also managed to complete 2 summer placements at different institutions, which were extra that I just emailed academics to ask about. So between my 2nd and 3rd year, I went to another research institution in London called the Francis Crick Institute and did a summer project there with a group called the Oliferenko Group. What you'll find is research groups are run by a particular scientist, a professor or a doctor, and their surname, so in this case, she has Hannah Oliferenko, her surname Oliferenko becomes the name of the group. So that's why you see lots of blah group, blah group. So my first summer project, I did the Oliferenko group. Then I did my undergraduate project at my dissertation project, the compulsory project, in a group at King's called the Cox Group. And then between the end of my undergraduate degree and my master's degree, I worked in the biochemistry department here at Oxford in a group called the Corman Group, doing similar work as I was doing in the Oliferenko group. Different project, but had connections, which is how I got the second placement. Then I just finished in September. I did my master's degree last year between 2023 and September 2024. Imperial College in London. I studied MRes cancer biology to focus on and move towards a career in cancer research, because that's what I decided on by that point. And as part of that degree, MRes means Master of Research as opposed to MSc. So it's got more of a research tilt. So a large proportion of it was research based, whereas in an MSc master's degree, you'd have lots of talk content instead. A smaller research component. So as part of that degree, I got to complete 2 research projects. The first one was actually at a different university. It was a collaborative project, so I worked in the Goot group at University College London. And then the second project I was far more interested in, and I got to work with a really great lab called the McNish group, or the McNish lab at Imperial. And then now I've managed to I'm now a DPhil student. So DPhil is just Oxford's fancy name for a PhD. So it stands for Doctor of Philosophy still. I'm now working towards my doctorate in oncology, so in cancer research within a group called the Blagden Group at Oxford's Department of Oncology this time, rather than their biochemistry department where I worked before. And as part of that project, I've also been very, very lucky in that my co-supervisor, the deputy supervisor, that will be teaching me a technique that will help me with my project, is actually at the University of Cambridge, which is Professor Lilly. So I get to work in my secondary lab for my doctorate is at Cambridge. So I've altogether managed to work at 6 different institutions, which is tough to do. And I'm very, very grateful for having the opportunity to do that. And it means that I know a lot about different research cultures now from working in different places. So I would really encourage anybody who wants to develop a career in research to try and move around a lot when you're young and to just apply to different places, just email different people, because if you work hard enough and you're really lucky, you persevere enough, you'll be able to get placements in lots of different places throughout your career and your educational journey. Research career roughly works. If I get all of it up on the screen, the details of the two different paths, European, American, British, I won't go through again. It's not that important really. This is more for reference for your own knowledge later on when you get to the stage where you're just looking at different careers. Gives you an idea of how a career in research roughly looks. Generally, you do an undergraduate degree, then a master's for a year. So the undergrad 3 years, master's for a year, then a PhD 3 to 4 years. Sometimes the master's is integrated with the undergrad or the PhD. Then you'll do these postdoc positions, which are short-term positions, similar length to a PhD. They're the ones that do most of the research in individuals' research group. Then eventually you'll become a group leader or PI, principal investigator. But again, lots of variation. This is just an average, a rough guide to give you an idea of what a career would look like. Okay, this is where we get to the meat of the presentation, really. So, what does the world of molecular biology look like? Because that's the world that cancer research is within. So, really, this is roughly how I would define it, and you could define it in all sorts of different ways. I would say that if you have biochemistry as the starting point, which encompasses, by definition, any chemistry that occurs in a living organism, you can split that in two ways. You can split that into a molecular or cell biology component and a structural biology component. And you're going to be one of the two. Now, as a cancer researcher, I fall into molecular or cell biology, whereas there'll be some cancer researchers that are more on the structural biology side. So I sit on the top one. So what's the difference? What do I mean by these two areas of biochemistry? Well, molecular or cell biology, I would say really is about studying biological pathways within and between cells. So that it will typically look like protein A activates protein B, inhibits protein C, activates protein D, sometimes just small molecule messengers between the proteins. And that's a typical pathway, and they'll diverge and converge, and you'll be using techniques to study the pathways and manipulate them, maybe if you're trying to treat disease with drugs. But you'll be looking at these biological pathways. And the reason I like that area over structural biology is you're using a really wide range of techniques, because this is a typical figure from a paper. I think it's from a paper about muscle research, and you can just see the variation and the large number of different types of graph and different types of data they've got to analyze a particular biological pathway. And that's an example of a diagram showing all sorts of different pathways going on in a biological system. In this case, it is a particular type of blood vessel, I think. So that would be something along the lines of the muscles lining an artery, blood flow on the inside, and supportive cells on the outside, something like that. And what you'll see here is just a word cloud showing just how many techniques we use. And that's why I chose molecular cell biology over structural, because there's just so many techniques you get to have the opportunity to use and play with. Now, structural biology is a bit different. You really only have 3 techniques we use, but they're very expensive techniques. They're ultra specialist techniques. They're so specialist that you will literally choose really one of the 3 or maybe 2 of the 3, and that will be the one that you use forever in your research. As far as I'm aware, people don't tend to do all 3. Cryo-electron microscopy, which is based on the concept of electron microscopy, which you'll have heard about in your A-levels, but varied slightly, as we'll see. NMR spectroscopy, and then X-ray crystallography, which for reasons that you don't need to know, can only nowadays be done in the UK in a particle accelerator, which is halfway between Oxford and London, called Diamond Light Source. And structural biology is determining the structures of proteins, basically. If molecular cell biology is looking at pathways, protein A axon, protein B axon, protein C axon, protein E, structural biology is looking at a smaller scale, an individual protein, what's its structure, how does it interact with other proteins or other molecules, how can we drug it? So it's got a more of a chemistry focus. So as somebody who likes chemistry, when I went into my biochemistry degree, I thought, I want to be a structural biologist. I then changed my mind completely and went, that will be really boring. I'll just be analysing data all day on the computer. I want to do the less chemistry-based area and go into molecular cell biology. This slide isn't very relevant, but deep learning has also been solve lots of structural biology problems, because you no longer need to, in many cases, use these complex techniques to work out the structure of a protein. You can just give the sequence of a protein, the sequence of amino acids, to this AlphaFold computer system, which won a Nobel Prize recently, and it will, through the power of machine learning through AI, will predict how that protein will fold, and it does it with such a high success rate. In many cases, you don't need to experimentally determine the structure of a protein. So that has come along and completely changed the field of structural biology in the last 4 years. Right. Fundamental versus translational research. This is where it becomes relevant for what I do. What is the difference? It's a really broad term, fundamental and translational. I like to use it a lot. I say that fundamental research is a 1 in a million type research, in that if you're looking from the perspective of a disease like cancer, is you're researching for the fun of it, and you're researching because you're interested, but without a particular focus in terms of, I really want to get this particular pathway into the clinic as a means of improving cancer detection or treatment. If I happen to find something that's useful, that's really great, we can then give it to other researchers, they'll progress it to the clinic, or it will just be so good that it will get to the clinic really quickly in some capacity. So that's your 1 in a million research. Nothing wrong with that. We need fundamental research. Often we don't know what we're looking for, and we don't know how things are going to benefit society. So and that is true for any research field. Translational research, I say, is when you have a particular path. So say you have a signaling pathway downstream of a receptor in a cell that is switched off in a cancer cell. Its activity is reduced, it's downregulated somehow. Maybe a protein's expression is decreased in that pathway. Protein is mutated, so its activity is less, something like that. You could say, well, maybe we can switch it back on, or vice versa. If it's overactive, maybe we could have a drug which binds to a protein in it and stops the protein from working to shift its activity down. And once you've found that fault in the disease, in the cancer, it's then about really pushing it, kicking and screaming towards the clinic, because there's so many hoops to jump through to get any new drug to the clinic. In terms of both getting it through the preclinical stages, i.e. cell-based models of the disease, testing that drug would work, that thing would work in an animal model of the disease, and then getting it through the 3 or 4 levels of clinical trials, early phase clinical trials, and people who really have no choice but to go on a trial because they've exhausted all treatment options. Then the phase 2 trials in a large cohort, phase 3 trials in a really large cohort, and then you get it to the clinic. And that takes decades. So it's about really pushing your potential drug to the clinic. And there's 2 things you can translate, really, at least in cancer research or disease research. One is a novel drug target, so a protein that you think you can drug to inhibit or activate a pathway to treat a disease, to correct something wrong in a disease. Or 2, if a protein or a molecule is overexpressed, its levels are higher in a disease than in healthy tissue, maybe you could use that as an early warning system of that disease. Maybe you could use that as an indicator that a patient is likely to develop the disease, or likely when they do develop a disease, it will be susceptible to a certain type of drug. So that is called a biomarker. So you can either translate research into diseases as a biomarker, an early warning system of a disease, or as a potential drug target. Of course, you have to get a chemist to make a drug to target it, so it involves a lot of collaboration, and it's potentially harder than using your aberration as a biomarker. But it's about really consistently pushing your thing through all those hoops, through all those tests, to make sure that it will work. It is robust, it's not going to lead to side effects until it eventually gets to the clinic. So I like it because you've got to have that belligerence to push stuff towards the clinic in the long term. How long have I got? I've got time. So this slide, this is just an example of fundamental research. This is a guy who runs the Francis Crick Institute, and this is an example of where research that was done for the fun of it, just because it's interesting, really, happened to identify a molecular fault that was so good we could target it with drugs really quickly and get it to the clinic quite efficiently. So in this case, Sir Paul Nurse was in the early, must have been in the 90s or the 80s, was investigating particular factors, proteins, which control the cell cycle. So the process through which cells grow through to divide. He was investigating that in a species of yeast called S. cerevisiae. Now, why was he using yeast, not human cells? Well, it's because yeast are what's called a model organism, so they are cheaper than human cells to buy and to use, and there's less regulation about using them so than human cells, because they're yeast. It's the same type of yeast S. cerevisiae is used to make bread. But it's also they're simpler because they developed earlier in evolutionary time than humans. So for each family of genes in humans, a yeast will have fewer. So if you can understand what a particular protein does in yeast, how a particular pathway works in yeast or another model organism, there are lots. There's fruit flies, there's frogs, there's all different things for different purposes, different types of research. then it will give you an idea of how that thing will then work in a more complex biological system. So the evolutionarily later one, the one that developed later in evolution in history, which will be humans, Homo sapiens. So he found a gene called CDC2 in S. cerevisiae, whose homologue, i.e. the version of it in human cells, is called CDK1. He found that in yeast, that it was a major regulator of the cell cycle, this CDC2. It promoted cells to enter the M phase, mitosis phase, where they divide, and the S phase, where their DNA replicates. Hopefully you will know those terms from your A levels. And because he'd found that CDC2 does this, and it's such a key driver, because there's loads of drivers of the cell cycle as in any process, but it's such a nexus point. It has so many things coming off of it. If you imagine a big signaling pathway diagram, it's like a spoke in a wheel in a big network. It's a big spoke in that network of signaling pathways in charge of the cell cycle. Because it was such a big spoke, he could target it really quickly. It really overnight changed the way we understand the control of the cell cycle. And now drugs, I think drugs targeting CDK1, it's CDK1 inhibitors, so things that block it to slow down the cell cycle and slow down cell division rates, are in the clinic to treat cancer. If they're not, they're in late-stage clinical trials. But the gist is, those drugs that target it got there really quickly once he found it, because it was such a good drug target, he found it by chance. And through this work, as a result of this work, he got a Nobel Prize, and he got a knighthood, and he set up and runs this big organisation, the Francis Crick Institute in London. And before that, in 2001, I think, he was also the man that came up with the idea of merging lots of the existing cancer charities in the UK to form Cancer Research UK. But again, all of this wouldn't have happened unless he'd happened to have found this discovery. So that's fundamental research, the 1 in a million idea, which is, and most research that doesn't end up revolutionizing the world. Most people don't go on to earn Nobel Prizes. Why I like translational research is you're not going to change, you're not going to cure cancer, but in one person's career and one person's lifetime, you can hope to get a marginal gain. You can hope to get a particular pathway that you notice is aberrant in a cancer cell, and you can hope to target it, or use it as a biomarker, and in doing so, get it to the clinic and have some kind of clinical benefit for patients, i.e. improve lifespan in terms of a drug, or improve lifespan in terms of a drug, and earlier diagnosis in terms of a biomarker, so that you can help patients. You're not going to change the world overnight, but you know that you've got a fairly good chance of having a marginal gain and getting something from your work. Whereas with fundamental research, it will probably just be abstract and remain in papers forever, which is fine if you really like that, but I really want to make my research have a real world impact in the long term. And that's not easy even with translational research, but it's all about a real push to get stuff to the clinic in whatever way you can. So you get some kind of benefit for patients, even if it's only tiny. So, this slide we can skip. So that slide just, you can look at it later. It just gives you an idea of the plethora of research projects you'll come across throughout your time as a student, student researcher, because these are all the titles of the research projects I did before I started my PhD, my DPhil on the top, and then that's the title of my DPhil project below, so you can read through them later, the titles, and just get a feel for how many different research fields there are. And they're all really in the world of fundamental research. The only cancer research project listed there is from my master's degree, which is this one about Schwachman-Diamond syndrome and ovarian cancer. Right, so now we've got the biggest part of this talk, which is the two translational oncology case studies, I called them. So these are examples of where somebody has identified a fault in a cancer cell, in a type of cancer that occurs in some patients with a type of cancer, and they've managed to target it, either by using a protein in that pathway as a biomarker and got that into the clinic, or as a drug target and got that into the clinic, and had some marginal benefit for patients. Now, actually, the first case study is a proper success story in that the ASS1 story, which is arginine-succinate synthetase, that's what ASS means, it's a particular enzyme, ASS1. They've managed, that enzyme is downregulated in certain types of cancer, and by targeting it, that scientist who discovered this has been able to get improve outputs for patients with that type of cancer. And the second story isn't quite a success story yet. It's my supervisor's research story, which she's found a protein called LARP1 or LARP1, which is overexpressed in early stage ovarian cancer, and she wants to use that as a biomarker to allow for the earlier diagnosis of ovarian cancer. Hasn't yet, but that's hopefully a success story in progress. But I thought they give you a good idea of the perseverance involved in getting these two molecular aberrations to the clinic. One of the drug target, the other as a biomarker, and hopefully they'll give you an idea of what translational oncology means and how one person's career can lead to improvements for a particular group of patients and can have a real world impact, which is what I'm really passionate about oncology doing. So the arginine-isocyanates synthetase story. So arginine, first of all, what is arginine? So arginine is this molecule here. It's an amino acid. You'll know there are 20 naturally occurring amino acids used in all proteins in your body. The sequence of amino acids in the protein determines the protein structure and function. Now, arginine-isocyanates, well, this cycle here is a particular metabolic cycle. So each of these words here is a particular small molecule, small chemical molecule in the body. The things in squares here are enzymes which catalyze the different steps of the metabolic cycle shown here. So this is called the urea cycle. It occurs in all cells of the body, but particularly in the liver. And one of its functions, because all metabolic pathways are linked and have multiple functions, one of its functions is to produce the amino acid arginine, which you can see here as one of the molecules in the middle of the cycle. So some of this arginine will be siphoned off to form proteins to do all the jobs that arginine does in the body. The rest will carry on, and these other molecules will fly off to do what they do in the body. Now, ASS1 is one isoform, so one version of the enzyme that sits here. There will be several ASS, I can't remember how many, there'll be several ASS enzymes that can catalyze the conversion of citrulline to this arginine-isocyanates molecule in the body on the way to producing arginine. Now, why does this matter? Well, so there are a particular type of tumours that technically aren't lung cancers because they form on the lining of the cavity in the body that the lung sits in. So if you imagine the lung sits in the body and it's got a cavity around that, and the lining of that internal body cavity is called the mesothelioma. Sorry, mesothelium. And the cancer, when the cells that form that lining of that cavity proliferate, it becomes mesothelioma. Now that you'll know because that's the cancer that is caused by asbestos. Not quite in all cases, but in most cases it's asbestos linked and asbestos caused. Now this particular researcher, he's a guy called Professor Peter Slosserak. He works at Queen Mary and the Barts Cancer Institute in East London. So he works towards the east, the city centre of the city of London. I can't remember the place now. And he found that in some mesothelioma cells, not all of them, but some, I can't remember what the proportion was, but it was quite a large proportion, the enzyme ASS1 was downregulated. Its expression was reduced compared to in healthy cell mesothelium cells. Reasons for why it's downregulated aren't particularly important. There's a couple of mechanisms. One is a transcription factor called HIF1 alpha he found that binds to the promoter of ASS1, the thing that normally regulates the transcription expression of that gene, and switches it off in cancer cells, which it doesn't really in healthy cells. And also this promoter was modified, so it had these methyl groups attached that blocked the expression of that gene as well. But the key point is that gene was downregulated, reduced expression, so reduced production of arginine in healthy cells, in mesothelioma cells, cancer cells, compared to healthy cells. So that's the molecular aberration he found in his cancer cells. Why did the cell do that? Why has the cancer cell developed it? Because whenever a cancer cell develops and conserves a particular molecular change, it's usually because it promotes it or drives it in some way. It's the evolution on a mini scale. Well, the reason why it had developed and conserved this downregulation of ASS1 in certain patients is that when you decrease the expression of ASS1, you get decreased conversion of citrulline to arginine-isocyanates, and so you get a build-up of the precursors to arginine, specifically citrulline and ornithine in the cycle. And one of them, I can't remember which one, can be siphoned off to form nucleotides that form DNA. So when you get a build-up of these, you get more nucleotide production because more of them are siphoned off to form nucleotides, and those nucleotides then form DNA, and because you've got more DNA, the cell can divide more rapidly, and it promotes more rapid cell division because the cell will go more DNA, must replicate more rapidly. So it's an oncogene, essentially. So it's a tumor suppressor gene. When it's switched off, it promotes the cancer, ASS1. So that's the pathway through which ASS1 loss promotes cancer. But this scientist, Professor Slosserak, said, well, how can we target that? How can we target this decreased expression of ASS1 in mesothelioma? Because it's not happening in healthy cells, so we can hopefully make a drug that's selective towards those cells based on the fact that only they have decreased expression of ASS1. So what did he do? So he developed a drug called ADI-PEG20 or PEG-arginase. Now, this isn't a typical drug in the sense that it's not a small chemical molecule that binds to a protein and stops the protein from working. Like, for example, paracetamol or aspirin. They're small molecules that bind and stop the protein from working. It's a protein itself, and that's becoming more and more common nowadays. You have proteins of various types that you either artificially synthesize, and they'll usually be modified forms of an existing, naturally occurring protein. You'll then extract them from your whatever organism, like a bacteria that's made them, and you will inject them into patients as a drug. So this one, it's a modified form of an existing bacterial enzyme called arginine deaminase, or ADI, and it's modified basically with these chemical molecules that just mean that when you inject it into patients, it lasts for longer and the body breaks it down more slowly than it would in an unmodified form because the body can less easily recognize it as foreign or bacterial. So it's called ADI-PEG20 or PEG-arginase. I prefer PEG-arginase because it sounds more like a typical drug name. And it's first in class, which is meaning no other drug exists apart from this one that works via the same mechanism of action, has the same mechanism of having effects on the body. Now, what is it? Well, arginine deaminase, PEG-arginase, is a drug which breaks down arginine. Essentially, bacteria can break down arginine to produce energy. Humans can't. Arginine deaminase is the enzyme that bacteria use to do that. I think it's bacteria, it's them or yeast, one of the two. So he tried it, first of all, PEG-arginase, as a monotherapy in patients with what cancer? Because it's not just mesothelioma he found, that's the one he's focused on, but not just mesothelioma had a decreased expression of ASS1, some subsets of patients with other cancers did as well. So to start with, he trialed it in a type of liver cancer, a type of skin cancer, and a type of blood cancer. But its efficacy, when you tried it in the trials, in terms of clinical outcomes for the patients in the trials, proportion of patients with an increased lifespan, decreased rate of growth or progression of the cancer, these kind of clinical statistics, its effect on those statistics, efficacy in these trials, on its own, so as a monotherapy, wasn't great. It was quite variable. And why was this? Well, it's likely because when they did the trials, they didn't subset the patients by whether they had decreased ASS1 expression or not. As in, what they did was, and I'm not sure why they did this, I'm presuming just cost efficiency, they went, well, large proportions of patients with these cancers have decreased ASS1 expression in those cancers, but not all of them in each case. We'll just try it in a random set of patients with those cancers, not checking if they were specifically patients with decreased ASS1 expression. So that's the most likely explanation for why it had variable efficacy across the trials, because it was most effective in those where the patient's cancers had decreased ASS1 expression, rather than not so effective in the ones where they didn't. So, because it has quite effective in some cases in monotherapy, they then put it into a larger phase 2 trial. And in this trial, it was called the Adam trial, and it's quite big news at the time for Queen Mary because they did get an improvement in outcomes, and it was a statistically significant, so not a random, not an outcome that was due to chance. It was likely a real increase in outcome. It likely has a real clinical benefit, and this drug, PEG-arginase, increased the median progression-free survival. So that it's a measure of how long it takes for the cancer to progress. So the longer the period the cancer doesn't progress for, the more effective the drug is, and it's used as a proxy for survival, because the greater the progression-free survival, the greater the overall survival will likely to be. But you're going to get the statistics quicker because progression-free survival will obviously be shorter than overall survival for patients. So they found that with PEG-arginase, patients with mesothelioma had 3.2 months progression-free survival, PFS, compared to 2 months for those that were given what's called best supportive care. So basically, palliative treatment, so weren't given a treatment. So that's great. It has a real impact. Not a huge one, it's only 1.2 extra months. But if you look, the patients already only had 2 months progression-free survival. It's a really kill you quick cancer. So it's not a very pleasant cancer. So even a 1.2 months increase in survival, brilliant. They then went, can we go even better? Can we get even more bang for the buck? Can we even get more of an improvement in clinical outcomes with PEG-arginase? So he did some more molecular biology work, some more lab work. He found that in the bladder cancers, he looked at bladder cancers this time that had decreased ASS1 expression. I think he was doing a collaboration with a bladder cancer scientist. That's why I looked at bladder cancer now, not mesothelioma. He found that in those cancers which were ASS1 deficient, decreased ASS1 expression, there was decreased rates of nucleotide synthesis and nucleotide uptake. What I mean by this, and that it's a bit confusing because I said earlier that when you have decreased ASS1 expression, you have increased nucleotide production. I don't fully understand this. I think what it is that those cells are trying to compensate for the fact that there is a greater push towards nucleotide synthesis in them to promote cancer proliferation. The cell is using its anti-cancer mechanisms that exist already to compensate for that and decrease the other aspects of nucleotide synthesis and nucleotide uptake, so they don't divide as much. So you still get an increased rate of proliferation compared to healthy cells that don't have that AS1 downregulation. But it's a compensatory mechanism. Anyway, not that important. The point is they found that when ASS1 deficiency is decreased, you have a second knock-on aberration in the cells, and there is, you can see here, these black dots represent the a signal that corresponds to the amount of protein in a particular cell extract, and you can see that when you add this pigargiminase to them, you can see these two different cancer cells, T53J and T24, which are two different types of bladder cancer cells. You have decreased expression of these two things, DHFR and TS in each case, and their molecules, that one's involved in nucleotide uptake, that one's involved in nucleotide synthesis. The key point is they found this second aberration, that we already have a chemotherapy drug that targets, chemotherapy drugs being really non-specific drugs that kill all fast-acting cells. But we know this type of chemotherapy drug called antifolates, this chemotherapy drug class, target nucleotide synthesis anyway. So this guy, Professor Slosrak, said, well, why don't we combine anti-PEG20, pigargiminase with antifolates, because they're going to target the two aberrations that occur together in cancer cells. It would make rational sense, it's logical, to target them both together. And I say rational because often you'll just try any combination of drugs in case you find one that works. Here they went, we're specifically going to try this combination because they'll likely work together, and we don't need to get the antifolates through clinical trials because we know they work on their own. So it's less of a barrier to get them to the clinic. With ADPEG20, you just need to focus on getting the ADPEG20 to the clinic. The antifolates are already there and they've passed safety checks already on their own. So they tried pigargiminase with an antifolate, one of these chemotherapy drugs called pemetrexed, and they also tried it with a drug called cisplatin, another chemotherapeutic that had a different mechanism of action that's used all the time with pemetrexed to treat bladder cancer. And using this drug triplet, because it's 3 drugs, they trialed that in ASS1 deficient mesothelioma. So this one was the biomarker-directed study. They specifically looked at cancers in which ASS1 was deficient rather than just all mesotheliomas. They did that, it passed phase 1 trials. They then did it in a combined phase 2 or 3 trial, and that was called the Atomic Meso trial. And the results of that trial were brilliant for mesothelioma, in that they increased overall survival for patients in that trial from 9.3 months to 7.7 months. And progression-free survival, they increased from 6.2 months to 5.6 months. So that's brilliant. And what you've done here, one arm of the trial, one half of the patients, or set of the patients in the trial were given adipem-cis, the pigargiminase plus pemetrexed plus cisplatin, the full triplet. The others were just given the best therapy for mesothelioma that's in the clinic already, which is just pemetrexed and cisplatin together, no pigargiminase. And when you add the ADPEG20, the pigargiminase to the combination, you get this extra couple of months median overall survival and median progression-free survival. So that's a real world improvement in outcomes for patients, because it's really hard to treat cancer. You know, patients are dying within 7 months, 8 months, and now they've got a chance of, on average, living an extra 2 months with that therapy. Brilliant. And because of these trials, pigargiminase has now been approved and is now in use in the UK for certain patients with AS1 deficiency, with certain types of mesothelioma. And this chart here, orange line represents a particular type of graph called Kaplan-Meier plot that they use for trials. The key point is that the orange line is just the number of patients in on the PEMSIS, so the existing therapy arm of the trial, alive at each point. So as you go along, more and more patients drop off, they die. But you can see that the blue line, which is on the adipem-cis, so they've got the pigargiminase as well in their system, the rate in which they drop off is slower. And you can see that there's a number of patients alive at all points, but especially at the end, is higher than on the orange line. So that just shows what we've shown down here: overall survival, progression-free survival, better for patients with pigargiminase. So that's a real-world impact that this drug has had. And when this Atomic Meso trial came out, it was such big news for Queen Mary that they then put out there were loads of newspaper stories. So this is one I saw in The Guardian last year about pigargiminase for use for treating mesothelioma, so asbestos-linked cancer, and that was really exciting to see in the news. So this is Professor Slosrak, and he's researching in the lab now, wants to go even further. So what they're doing in the lab now is looking at the mechanisms of resistance that develop in cancer cells when you treat them with pigargiminase. There's a couple of ways the cells do that. One is antibodies, immune responses develop against the injected protein. Makes sense. You're injecting a foreign protein, the body's going to respond to it. 2 is tumors can switch back on ASS1 in response to targeting it. So that's kind of obvious, but that does happen. And another one, a more interesting mechanism of resistance that happens in more cases than the other two, I think, is that immune cells will actually be promoted by the tumor to come in to the tumor when you treat the tumor with pegARGINA. They'll start behaving abnormally, and through a complicated mechanism, they will add arginine back to the tumor, which is bad. Doesn't matter how, and they're trying to find ways of targeting that now. Now, in the 15 minutes we've got left, I'm going to go through the second success story in progress. It's my supervisor story of the way she wants to get a molecular aberration in this time ovarian cancer into the clinic. And that aberration is overexpression of a particular oncogene called LARP1 or LAR-related protein 1. And this is what I spend all of my day doing at the moment, and I will do for the next 4 years, is studying LARP1 in ovarian cancer. So, what's this story? So there is a particular type of ovarian cancer called high-grade serous carcinoma of the ovary. Don't worry about what that means. I will just refer to HGSC throughout the rest of this talk because that's the proper name for them. This subtype of ovarian cancer. It's the most common type of ovarian cancer. So when people talk about ovarian cancer, they will almost always mean HGSC. Not always, but almost always. And it's also the most lethal. It's the one that kills patients. The survival statistics are most dismal for. It's got worse treatment outcomes for. So what are the properties of HGSC? Well, as I said, they're most frequently diagnosed and lethal ovarian cancer subtype. If you look at the molecular aberrations that typically occur and conserve between patients, what are the most common ones? Well, frequently there are lots of changes in the gist. I won't go through them all. It's not very important. The key is there isn't a great deal of conservation. One of the most easily targetable is this type of DNA repair pathway that is switched off, down-regulated. In various ways due to gene mutations, inherited, or ones that occur randomly in the cancer cells, but not in the rest of the cells in the body, or because there's reduced expression of genes. That happens in about just over half of patients, and you can target that. We can't, yes, this pathway called the RB1 pathway, down-regulated in lots of cases, but we can't target that at the moment. There's no drug that exists that targets it selectively, so that's useless to us in the clinic. And the PI3K or RAS pathway, it's overexpressed, upregulated in about 45% of cases, but again, no real way of targeting that. The cells develop resistance to drugs targeting it really quickly, so that's useless in the clinics. The only way really we've got targeting it selectively, i.e. without just using chemotherapeutics that have lots of side effects and aren't that great, is to target this type of DNA repair pathway, which they do. You also have mutations in a gene called TP53, which occur in like 99.9999% of cases, and they happen at a really early stage. It's most likely the first event that leads to the development of a cancer, because cancer develops in the progression. And that's probably the first one. But lots of the details of how ovarian cancer develops aren't very well known at the moment. So, really hard to develop selective or targeted drugs treating HGSC due to the pathways that go wrong to drive its progression and its development. So, what is LAR-related protein 1? Why is my supervisor, why am I interested in it? So it's a really highly evolutionarily conserved RNA binding protein. So an RNA binding protein does what it says on the tin. It's a protein that binds to RNA and regulates some aspect of its functionality. So either drives its increased translation to get more protein synthesis, or decreases its translation to get decreased protein synthesis and decreased gene expression, or it could stabilize it, so you get in the long term more protein synthesis, or destabilize it, or just change its localization in the cell. LARP1 is a particular type of RNA binding protein, does particular stuff that, to be honest, we're still trying to work out what it does. Now, in healthy cells, it has a really, really complicated and poorly understood role in regulating, and it regulates the expression or translation, or a protein synthesis from a particular set of mRNAs in the cell, which encode proteins involved in translation itself. So these will be ribosomal proteins, other RNA binding proteins, translation factors that work with the ribosome to translate mRNAs. So because of this, LARP1 becomes this really big hub in the cell for regulating downstream protein synthesis, in that LARP1 will regulate the translation of mRNAs that encode ribosomal protein and others. So when you increase LARP1 expression of those mRNAs, you increase ribosomal protein expression and thus increase overall protein synthesis in the cell. That's a bit confusing. It doesn't matter too much. The gist is it's a really big signaling node involved in regulating protein synthesis in the cell through acting on a particular type of mRNA. And we really don't know what it does in those healthy cells still. And I say it's complex. When I say it's complex, I I spent all of the last weekend reading 30 papers which argued between themselves from different research groups over the last 5 years about exactly what it does in healthy cells. Some saying it upregulates expression of the transcripts, some saying it downregulates, and some saying it stabilizes, all different things in different papers. What does it do in cancer? Well, in ovarian cancer, in HGSC, it's overexpressed. And that overexpression we have shown in the Blagden lab. We're the only lab that looks at it in ovarian cancer, apart from one paper that was published recently by a Chinese lab. Other than that, we're the only lab that looks at it in ovarian cancer. And we found, my supervisors found that when it's overexpressed in HGSC, it happened at a really early stage, and it does definitely drive cancer phenotypes. So when it's overexpressed, it's correlated with adverse outcomes, which I mean the higher the level of the expression, the poorer the clinical outcomes of patients, the higher the level of the expression in ovarian cancer patients, the worse, the shorter their overall survival time, etc. when you look at clinical trial data and you look at their LARP1 expression measured in cancer patients, specifically in their tumours. We've shown that it drives many different cancer phenotypes. So it has a big role in driving the cancer because it doesn't just do one thing, it does lots of things. So it promotes cell migration and invasion, promotes anchorage-independent growth. So that's growth when the cells aren't attached to any other cells, which normal healthy cells don't tend to do unless they're blood cells or immune cells. So that's a phenotype associated with cancer, is that cancer will abnormally start replicating lots even when it's not attached to other surfaces. LARP overexpression drives increased cell survival, it will drive chemotherapy resistance, and it will drive the formation of these stem cell-like populations that have got, a lot of plasticity, they can respond to a lot of stuff. That's a real buzzword in cancer, it has been for years, stem cell-like populations, in that they're a really novel concept, in that cells take on these plastic properties in cancer. Don't know how targetable stem cells are they. And most recently, and we're trying to get the paper published at the moment, my lab has shown that LARP1 has a really big role in driving changes to tumour metabolism. And specifically, my lab think that the most of its cancer phenotype effects likely stem from its role in changing tumour metabolism. So by shifting major metabolic pathways up or down, regulating them in these ovarian cancer cells compared to healthy cells, we believe that it's then the knock-on having all of these other effects, because it's unlikely that one protein will act on so many pathways it has the effects independently. I've listed here. We think that the metabolism changes to that are what then leading on to these other effects like increased survival, drug resistance, etc. So my supervisor has also shown that LARP1 overexpression doesn't just occur at any old time point in HGSC formation and progression. It occurs at one of the earliest stages, and like I said, we don't know much about how ovarian cancer forms. We've shown LARP1 expression occurs almost immediately after the TP53 mutations that I said are the initiating event, or most likely the initiating event in HGSC tumorigenesis. LARP1 overexpression occurs right then. We got that from both mouse models of HGSC development and from patient biopsy data, where we looked at their existing pre-cancers, the precursors to their cancers, and found really high LARP expression in really early pre-cancers. Do we know the mechanism through which LARP1 changes metabolism to have these other phenotypic effects? Not entirely. We've got gist of it. We know it must be through having a different effects on our RNA because it's an RNA binding protein, or that's most likely how it will have effects to promote cancer. We're still trying to work out lots of details for how. We know that in cancer cells, in ovarian cancer cells, it binds to a much wider range of transcripts than just the set of transcripts that encode protein synthesis machinery components like ribosomal proteins, as it does in healthy cells. It binds to them in cancer cells as well, regulates their translation, just like it does in healthy cells. But it also binds to so many more transcripts, including cell survival factors, which is one of the ways it has effects on cell survival, and like I said, loads of metabolic transcripts, which we've just found. And the current theory, the current model in the lab, which is a bit vague at the moment, is that maybe LARP1 has two roles, evolutionarily conserved roles, and it's been conserved for so long in evolution, maybe that's because it has two roles. One, it regulates overall protein synthesis through acting on that set of mRNAs I said before in healthy cells. But B, it has a secondary stress role activated, usually transiently when the cells are stressed, where it acts on so many more transcripts that have all the other effects on the cell. But maybe in these ovarian cancer cells, because of the changes in the environment that occurs when the cancer develops, because of LARP1 overexpression, that stress role is really overactive. So rather than us not seeing it at all in healthy cells, we suddenly see it all the time, and just that in the unhealthy, the cancer cells, and maybe that's how it's having its effect on cancer. Still trying to figure this out. And my PhD project this title here, and it's looking at an aspect of how LARP1 overexpression in cancer cells drives these different cancer phenotypes. It doesn't matter how, you can ask me questions about that at the end. The key point is why do we care about this? Who gives the monkeys about LARP1 overexpression in ovarian cancer? In translational oncology, you want to ask the question, how is this going to have a real world impact? It's going to have a real world impact, potentially, and we're hoping, because of RNA Guardian, which is my supervisor's spin-out company. So it's her company developed just to translate this LARP1 research, where they've said, okay, LARP1's overexpressed in ovarian cancer compared to in healthy tissue. Now, often when a protein is overexpressed, the cell, and especially when cells go wrong, they'll be quite leaky, that protein will leak into the blood and the extracellular space. And then you'll be able to measure increased levels not only in the cells, but in the blood, which is much easier to access than the tumour itself. You don't need a biopsy, you just need a blood sample. And so she'd said, well, if it's overexpressed in the tumour, is it overexpressed in the blood? Is its levels higher in the blood at an early stage of ovarian cancer? Because we've said it's really overexpressed, not just at any point in ovarian cancer development, but right at the start. So can we detect increased levels in the blood and use that as a really key early warning indicator of ovarian cancer? Now, why do we care about that? Okay, well, firstly, you can tell a patient if they're going to get the cancer. But for all the money and the effort and the belligerence you have to do to get any molecular aberration into the clinic, the drug target, or in this case, a biomarker, just get it in there to tell people they're going to get a cancer sooner isn't that useful. All you're essentially telling them is you're going to die quicker. It doesn't help massively. Biomarkers are only really useful when they tell you earlier you're going to get the disease, but they also, in doing so, allow you to treat the patient earlier or treat them in a different way to the way you would normally, i.e. allow them to stratify patients for a particular type of therapy so that overall they get more benefit from that therapy and there are increased and improved clinical outcomes. So in this case, what you have, a bit of background of ovarian cancer, is that often women have inherited mutations in that DNA repair pathway I said previously, that mean that pathway fails in their body, and if there is an increased, therefore, chance they'll get the ovarian cancer. In women which those mutations are detected, in women with significant histories of ovarian cancer, and those mutations cause breast cancer as well. So often if they've got breast cancer at a young age, they're at high risk, they'll be sequenced for those mutations. If they have those mutations, surgeons will then, as a bog standard, will usually just take out their ovaries and their fallopian tubes and lots of their anatomy to prevent cancer forming as a precautionary measure. And that's really invasive, as you can imagine. It has lots of horrible side effects in terms of hormonal changes, including one of the side effects is death. So if we can use LARP1 as an early warning indicator by measuring its levels in the blood of ovarian cancer, the hope is, and this is the purpose of RNA Guardian, and basically my supervisor, Professor Blagden's life's work, is if she can detect LARP1 early, you can say to patients with those inherited mutations, okay, you've got the mutation, but we're not just going to chop out your ovaries straight away with all these side effects. We're going to measure over time your LARP1 levels in your blood, and only if they rise above a certain threshold will we say, okay, your risk of ovarian cancer is now so great we need to chop out the ovaries. And that way, hopefully, you can stratify those patients with those inherited mutations, and there are a lot of them. Hopefully, you'll be able to stratify them and mean that less of them, only the ones at most high risk of having the cancer, have to get those surgeries. Now, obviously, if we did a clinical trial and found that all patients with those mutations had such high LARP levels, they need to have the surgery removed. It's dead in the water, the idea won't work. But we're hoping that when we get LARP1 to the clinic and it gets into clinical trials, it will function, it will help stratify patients and mean that women with those mutations don't automatically have to have surgery. They only have surgery if they're the upper threshold of risk with those mutations. And that's how we're hoping to get LARP1 to the clinic and have a real clinical benefit for patients. And you can read more about that online because my supervisor is really big on media and does hundreds of news interviews like this one here. She was in a Spanish newspaper earlier in the year as well. And she's also set up a society called the LARP Society that specifically looks at all LARP proteins, the whole LARP family of proteins in a whole range of biological contexts, because she's really enthusiastic about LARP proteins, and that hardly any researchers do that, and that shows her enthusiasm for it. And also you'll find in the media that we're a really translational lab, we're really focused on getting stuff to the clinic, and we've now also started in the last 3 years, and which has kind of taken over what the lab is known for, even though it's done LARP for about 10 years or 20 years, is this thing called LungVax, which is the first preventative vaccine for lung cancer treatment, and is going to be part of a family of preventative vaccines funded by Cancer Research UK, a Spanish charity called CRIS, and the pharmacology company GSK, GlaxoSmithKline, longbacks, lynchbacks, brachyvax, and something else I can't remember. And the gist is we're in charge of longbacks, and they're preventative vaccines. So you'll see lots of that in the media as well, and that's really interesting to read about, but I don't have time to talk about it now, because that's not the area of research in the lab I'm focused on. I'm doing the LARP stuff in ovarian cancer. But again, translation. It's all about how we can get clinical benefit, how we can keep pushing stuff towards the clinic. Not only because that's how you get a fulfilling career, I think, in research, that's what I love about it, but also it's really cool that you can have a real world impact. And the whole point of this presentation is to show that you can have a real world impact and science doesn't have to stay abstract.